Oh, we're live? We're live? Oh, so we're running late. I think this is 2 weeks in a row we're late now. But hey, you know, at least we're consistent. Oh, no.
Well,
Rosa, technical difficulties, Rosa.
guys, it seems like we'll reconvene shortly.
Screw it, we'll keep talking.
Is it gonna
so,
come on?
It's
I
terrible.
think we should get a restart.
Yeah, let's get a restart.
No, I don't think you got first time. I think we're still live.
Yeah, we're still on.
Okay, we're still
We're
live.
still on. Yeah, we're still on, baby. So we're having some technical difficulties with the projector in the room. So sorry about that. Computer issues are fixed. Sound issues are fixed. So you know, at least it's a step in the right direction.
You know what? I think we're gonna switch to the whiteboard. We haven't done a whiteboard show since like episode 4.
All right, yeah, let's do the whiteboard.
Let's do a whiteboard.
All right.
Are we just gonna remain live in real time?
Yeah, why not? Hey man, this is an easy peasy show. When life gives you lemons.
Maybe citric acid.
Yeah. So if you can, while we're going through this, post up the pictures of the slideshow on the live. All right, everybody. So sorry for the technical difficulties.
I can't because my Facebook situation.
Oh no. Oh jeez. Hold on. I think we're back to it. Well, let's keep this off to the side, just in case. Sorry, everybody. You get to watch us fumble around. All right. So, welcome to episode 18. We like to have fun around here, you know? We just like to...
We need theme music. Like, you know when the newscasters come on, it's like that.
Yeah, something like that. Rosa, can you like do a little fun?
Music 7, positive publicity.
All right, so guys, welcome. Episode 18, What is Cancer? Joined by Lebaron Agostini.
Greetings.
And obviously, you guys know me, I'm Dave, or David Deming. We're going with a nice, clean look now. We're going to start dressing appropriately for the magnitude of discussions that we have.
Yeah.
So,
I had a photo shoot earlier, so shout out to my photographer.
that's bougie right there.
But yeah.
All right, cool.
So what is cancer? What is
cancer?
What is going on, Dave?
So cancer is uncontrolled growth of cells in the body that most people know as a tumor. However, all tumors are not cancer, right?
Right.
Yeah. You can have a cyst, you can have warts, you can have different fibroids.
Right.
Fibroids. And which is, a cyst is just a closed pocket of tissue. Pus, a tomato.
Tomato, potato.
Oh,
Yeah, you know.
I like potatoes. No, I'm talking about pus.
Right.
So, cancer. How do doctors and scientists define it? They define cancer by the hallmarks of cancer. And that is based upon a paper that was written by Douglas Hanahan and Robert Wienberg. I see that Robert got cut off. Oh, sorry, Robert. And that was presented in 2000, and that was like the first generation of, like all the ideas the research field and the clinical field used to define what is cancer. And basically what we're going to do is we're going to use that as a template for tonight's talk and go through each individual layer of the complex issue that is cancer. It's not just one disease, as everybody, like a lot of people in the general public think cancer is one disease.
Yes.
It's think about it, it's thousands of diseases.
Well, I like to use this analogy. Cancer is like automobile.
Yeah.
You got Lexuses or BMWs or Mercedes. You know, there's similar things that all unify them. They all have 4 wheels. You know, they're all made of aluminum or steel. But parts from Mercedes-Benz aren't going to necessarily work in your Toyota.
Yeah.
And cancers are very different in that sense. Yeah, they're all cells, but their origin, the reason what turns them into tumor cells, the way they grow, the way they proliferate, have many different factors, and that's actually what makes treatment so hard.
So basically what would cure or treat one type of cancer could actually aggravate or make another cancer actually more virulent or more aggressive, more malignant.
Right.
And that's really my hang up with all the pseudoscience out there and all the big pharma is keeping the cure to themselves because
it's actually really lucrative to cure diseases. shout out to Sovaldi real quick, because that drug cures hepatitis C, and it's just as profitable as like Pegasus or like any other hepatitis C drug.
It's actually really expensive. It's like
super expensive.
$60,000.
Yeah, actually, I think the range is like 60 to 100K depending on like your disease state. But yeah, that's people aren't hiding cures for you. We literally work to find cures.
And the thing about cancer is you can cure, if you have the cure for cancer, it's not like it's not going to come back. Like that's how our bodies adapt. Cells adapt to any type of input that you send at them. So let's say, chemo. You blast something with chemo, you blast something with radiation, those cells that live are going to be stronger in the fact that they didn't get killed by the chemo. So they're going to grow, they're going to divide, and they're going to reproliferate and make up the tumor again. So that's what, what's called
relapse.
Relapse, yep.
Yeah, I mean, a lot of the times, that situation is dependent, of course, depending on like the oncogenic event that gave you cancer to begin with. Mm-hmm. If you have some somatic mutations in which, you get cancer, you get tumor, and it's possible for you to kill all those tumor cells, not necessarily. You do, when you use anti-neoplastics, you do select for the cancer cells that are resistant to whatever drug you're using.
Exactly. So you're talking about like such things as like tyrosine kinase inhibitors, like
right. But there's some and you have resistance. You do have some resistance, and you select for that resistance, just like how antimicrobials select for antibiotic resistance when they're used.
I know that's a big thing that's coming up. We should talk about that in an episode as well. Yes. Hallmarks of cancer that were defined in 2000 by, again, this paper in Cell. Sustained proliferative signaling, evading growth suppressors, activating invasion and metastasis, resisting cell death, inducing angiogenesis, and enabling replicative immortality. So those are just 6 generalized areas that encompass, and actually they overlay on each other if you think about it.
Yeah, a lot of the times.
And they intersect. So let's dive in. Sustained proliferative signaling. You could think about it like this: cells need signals or stimuli, environmental stimuli to grow, to populate, to split into 2 different cells. So when a cell divides, it splits into 2, 4, and 8, and 6, and 16, 32, 64, blah, yada, yada, yada. But you need things like growth factors, survival factors, such as IGF-1, insulin growth factor 1, chemokines.
B cells.
I have 7 B cells,
yeah.
Hormones, and these are just, think of them as like chemicals or proteins that tell the cell to do what it needs to do. And sometimes these pathways that we can see behind us get rewritten, or they get turned on all the time. So basically what that does, it tells the cell to grow and proliferate and rapidly divide much quicker than it actually should in comparison to its neighbor cells that maybe say are normal, that aren't cancers or not tumor cells. So they outcompete and they outgrow the cells that are around it in the tissue, and thus you get a tumor.
So the interesting thing is that when we get into an invasion of apoptosis later, the default position of cells is apoptosis. Mm-hmm. If you take away all the stimulus for cells, they will die. Right? There are constant signals that are going on telling the cells not to die. Mm-hmm. Right? So cancer cells want to maximize the signaling effort. Right? So this is how they find ways to turn on those signals constitutively, or what we say all the time. Mm-hmm. Right? And by allowing these things to work on their own, this constitutive signaling without that stimulus allows those cells to become anchorage dependent, to just be pretty much self-sustaining as far as the growth factor signals are concerned.
Actually, we're going to talk about that in the next slide, anchorage independence.
Oh, that's
contact inhibition.
Segued right into that.
Perfect. So basically, these hormones, like everybody knows about testosterone and estrogen, and in particular, prostate cancer and breast cancer are vaguely affected by those. If you have the androgen receptor in prostate cancer, can actually be turned on always, and that's what we call constitutively active, even if there's no androgens in the body. So you can go through what's called castration therapy, and it's either, it used to be surgery, but they moved on to a chemical castration. Yep, snip, snip, snip.
Yeah, it wasn't a heat siren. That was a
snip, snip, snip.
desactication over there.
Oh, Jesus, that sounds terrible. But it basically turns on the AR receptor, the antigen receptor, and it makes the cancer cells proliferate faster and faster and grow and more and more and more. And it doesn't necessarily mean that a receptor has to be turned constitutively active. It could be a downstream signal from that. So let's take the MAPK kinase pathway.
Where I have an analogy for this.
Nice.
Yeah, I have an analogy for this, right? So for example, say that turning the doorknob created a signaling pathway, right? Somebody from the outside turns the doorknob, and that tells somebody on the inside to come and open the door for them, right? Let's just say how that system works. Now, say the inside of the room is a cancer cell. Now, if you, the inside of the room, the cancer cell, found a way to produce that same stimulus without somebody being on the other side of that door, right? So they found a way to propagate that signal internally without someone being on the outside. That's pretty much how these MAPK signals work. There's no outside stimulus, there's no receptor stimulus, but the intercellular pathways are still being activated. And that allows the cells to do pretty much anything they want.
That's pretty
In regards to proliferation and activation of pathways.
interesting. So it's basically, you have a nosy Nelly in the middle of your whisper down the alley, and it keeps whispering and whispering and whispering.
What?
Yeah, you like that?
What reference is that?
I don't know. Just whisper down the alley.
I think
so
this
you go from one
must
person
be a
to person.
childhood Philadelphia reference.
What, you've never done whisper down the alley?
I have no idea what whisper down the alley is.
So you literally
That
have
sounds like something we're not supposed to do in Brooklyn.
10 people sit, since you get stitches. So you get 10 people sitting in a row, and you start off, and I go, I tell you a secret, and then you tell the next person, tell the next person.
All right, so that's telephone.
Oh, you call it telephone?
Yeah.
Okay, yeah, we call it
Whisper down the alley.
And it was Lehigh Valley, so I don't know what they do in Philly. What do they call it in Philly, Rosa?
Whisper down the lane.
There you go.
I guess. Wow. Please comment your whisper down the alley
Yeah,
experience from wherever state you're from. That'd be interesting to hear.
Yeah. So it's just that I always thought of it as a rewiring. So you have a light switch and it just gets flipped on, and you can't, even if you unflick the switch, it's still on. Lights are still on.
Well, mutations definitely play into. So when we're saying that cancer cells figure out ways to do this, 99% of the time they figure out ways to create this sustained proliferative signaling via mutations in proteins that are required for this signaling.
Yeah, it's definitely true. And a lot of them, which is what, KRAS, the RAS family is
The whole
definitely
MAPK
kinase. MYC is overexpressed in a lot of cancers.
P53 is knocked down.
P53 is knocked down. RB is knocked down.
ARF.
And ARF.
We could just name all these. Yeah, I mean,
all these different signals, and you can think of proteins in the cell as different nodes of a network. So one protein connects to another protein, that protein connects to another protein, and they're all interconnected. So it's not just like one protein connects to another, to another, to another. That'd be too simple, that'd be great.
Right.
But these proteins interact with one another and relay signals. So if you have one that's screwed up, it's going to tell 10 proteins to go and do something that it shouldn't be doing. And that's how you get sustained proliferative signals.
yeah, like the Philadelphia chromosome positive fusion protein.
Mm-hmm.
So that's a kinase that phosphorylates a whole bunch of proteins, including cyclodependent factors, and you get sustained proliferation through phosphorylating or activation of intracellular proteins that normally wouldn't be activated.
It's true. So the next category that we're going to talk about is evading growth suppressors. So there's not only signals that tell cells to stay alive, there are also signals that tell the cells to kill themselves, literally, to go through what's called like a self-destruct sequence, or apoptosis. And a lot of things that we just listed, like P53, RB, ARF, these are proteins that regulate the cell cycle, or they monitor DNA damage. And if you have an increased amount of DNA damage for whatever reason, whether that's an oncogenic mutation, or if it's, the cell just gets damaged, and these proteins don't catch that, propagates, and they can evade this type of signal.
And something that we mentioned earlier with the anchorage independent. So normally, very few cells in your body are in non-contact with each other. Most of the time, cells are in contact with each other, and that connection, that stimulus, that anchorage, is a signal that cells need, right? So you remove that signal, cells usually die because of it. Cancer cells, though, have programmed themselves in such a way that they don't need that anchorage anymore. Actually, one of the hallmarks of cancer, one of the things that we say is unique about cancer cells is that they're anchorage independent.
Mm-hmm.
This allows us to culture them by any means, and they just won't die. I mean, they'll die if we purposely kill them, but just alleviating that anchorage won't allow them to die.
Just like everybody says, oh, cannabinoids kill cancer cells. Bleach kills cancer cells too.
Bleach in a petri dish, yeah.
I'm just saying. I don't know what I'm saying. I'm just saying what I just said. But, I'm recommending that if you believe that cannabinoids are the cure for cancer, or there is a cure for cancer, bleach a quart of it every day. It'll get rid of all the cancer cells you have.
It'll get rid of all of the cells that you have.
Let's be really honest. You will die if you drink bleach.
Yeah, for those that didn't catch the level of sarcasm, so please don't.
Please
don't go home
God.
and just you and your Clorox just get real friendly. We don't want that. So, yeah.
Yeah,
Sarcastic, please.
But,
I think this will be our first lawsuit. I'm just like, Jesus.
Thank God we have an LLC.
Right. So evading growth suppressors.
ARF. Actually, ARF and P53 are the most highly deleted in immortal cells.
Yeah, of course. So pretty much P53 is a DNA binding protein that turns on other genes, right? And usually it turns on other genes that tell the cell to stop growing or to die. Those we either call them cell cycle arrest or pro-apoptotic genes, right?
Mm-hmm.
Cell cycle arrest means the cell is proliferating, P53 gets activated, all of a sudden the cell stops dividing. It doesn't necessarily die, but it stops its division cycle.
And that's what's called senescence, so escape from the cell cycle. And these proteins get mutated as well. What is it? 50-some% of
yeah, something like 50% of all tumor cells have mutations in P53, either partial deletion, mutations that change its binding site, null mutations, and there's actually a growing field that shows that mutations in P53 have different effects. They even have pro-tumorigenic effects.
We just had that article in Journal Club that the truncated version of P53 and exon 6, I don't really believe in garbage, trash paper. But
there have been shows that some mutations in P53 can play a role in, or can do things that even wild type or normal P53 can't do. And even different post-translational modifications of P53, like acetylation of lysine 120, can change the DNA binding effect of P53 and thus changing the way P53 regulates the cell. So all these things are very important in regards to
And to connect the two ideas that we just covered, it's switching from a growth suppressor to a more proliferative signal. So it's telling the cells to grow and divide and become, grow, like literally multiply. And then if you have that happen, you're gonna get cancer.
Yeah, unfortunately.
So that's, so there's a complexity to this. So it's not just necessarily one thing does this all the time and you get no change. Right. So that's basically why it's really difficult and it's nearly impossible to have a cure for cancer.
Yeah, I mean, the total complete. Again, we're talking about a multitude of diseases that have similar characterizations that we say, that we define as cancer. But the events that cause these cells to transform into cancer cells are very vast. A multitude of things can cause a normal cell to turn into a tumor cell. Mutation is just one thing, and that can happen in any number of genes.
Yeah. So I put in this nice graph or chart, not necessarily a schematic, of the extrinsic intrinsic pathways to apoptosis. So extrinsic is where you see where Jimmy Carter actually got this PD-L1 treatment. I forget the actual drug's name. Do you remember the PD-L1?
No.
So basically, he had metastatic melanoma. Yeah. And basically, what that does, it activates the extrinsic pathway for apoptosis. So cells can be killed internally, so they'll have internal signaling, or they can have a signal from the outside, whether that's TNF alpha, tumor necrosis factor alpha, or FASL, FAS ligand. Actually, do you know what FAS means?
FAS?
Yeah.
No, I know it's a death receptor signal.
It is a death receptor signal. And that basically sends its signals to caspase 8, and and then that'll actually activate BID, knock out VAX, or block cell2, and VAX will Keytruda.
Keytruda is
So Keytruda basically acts as a means to.
That's the brand name. The chemical name is Promprosimab. God, Jesus, I don't know. Promprosimab.
Yeah, it's an antibody.
This thing's got like 17 letters in it. Jesus, I don't know.
So that'll activate the same pathway downstream as the intrinsic pathway. And these can get hardwired in wrong ways. You can have BCL2 mutations, you can have, I know PARP can be mutated, and then the intrinsic can activate through just internal signaling, whether that's P53 or some other proteins, and basically activate caspase-3. Caspase-3 comes and just starts blowing stuff up.
And so these are proteins that regulate apoptosis, which is programmed cell death. You modulate their activity, or you remove some of them, and that changes the way a cell can die. Apoptosis is a normal cellular process. So
it can be dysregulated, just like we said, with proliferative signaling and the growth suppressors. So growth suppressors will actually make your cell grow for apoptosis. But even if you do have your P53, you can have other mutations in other proteins that will block its function. And will actually keep it from actually going through apoptosis.
Like MDM2 or
you like MDM2, don't you? That's a protein,
man. I like
So
Mick.
Another thing I wanted to point out with the chart on the right, left, I don't know, this side. I don't know, I'm looking at it.
Right hand side.
So the right hand side. So you can have treatment, let's say whether it's chemotherapy, radiation therapy, what we had just said with the Keytruda, crizotinib, erlotinib, all the ABs and the IMs.
Yeah.
You can basically have these signals being pushed into these cells, and the cells will have a subpopulation. Like, let's say we have 100 cells. And then, let's say 5 of those cells are resistant to anything you throw at it. So they have armor, they have shields, and they can block whatever the heck you throw at it. Those 5 cells will outlive, the other 95 will die, but those 5 cells who are resistant to the therapy will repopulate the growth, repopulate the area. So therefore, the next time you throw this therapy at it, it's not going to work. They already have a natural defense mechanism against it. Or they're rewired so that you actually can't do anything to it. So again, even if you do come up with a cure for a particular type of cancer, there might be a subpopulation of cells that...
Yeah, I mean, we see this with people that are diagnosed with childhood cancers. They have a higher chance of recurring cancers, most likely because whatever genetic instability was associated with a reason for getting cancer in the first place. Also, anti-neoplastics, 90% of them work on DNA damage. Cancer cells are more susceptible to DNA damage because they're replicating faster than other cells.
So they'll be in the S phase.
Right. So, and because of that, if you create enough DNA damage, the cell will reach a threshold in which it can't rescue itself, and the cancer cells will die. But part of the problem is that there are some side effects associated with using DNA damaging agents to begin with, and part of that increases your risk of having cancer post-treatment. And this is why people have these side effects where they're losing hair, they have gastrointestinal issues, they have skin issues, they have immune dysfunction, because all these cells are rapidly dividing, and a lot of these drug targets are non-selective.
Yeah.
And part of the reason why we study cancer biology is so that we can look at the specific details and orientations of the cancer biology to make drugs for those.
And instead of using maybe like chemo, I think it's like a bazooka.
Yeah,
It's an RPG, but we're trying to find a sniper.
Yeah, exactly. I mean, drugs from the 80s and the 70s, 80s, and 90s was a shotgun approach as far as it came to killing cells. The whole immunotherapy, the whole monoclonal antibody, this is more of a specified sniper type approach to try to kill some of these cancers. But we find that there's new problems with that, and no treatment is perfect.
Like I know that EGFR in non-small cell lung cancer, you would hit it with erlotinib, but erlotinib only targets the outside portion of the EGFR receptor, and the mutation is actually on the inside. Okay. So if you have different EGFR mutations, particularly for different types of lung cancer, specifically one subtype of lung cancer, targeting it with that particular antibody has no effect because of the type of mutation. So these proteins that we're all talking about can have several different types of mutations that cause them to be turned on all the time.
It's just like a triple negative breast cancer. You know, and so the negative comes for, so you never heard of HER2 positive or ER positive. You know, there's the estrogen receptor positive, or so if you, and these means that those type of receptors are on the cell, right? If those types of receptors are on the cell, that tells us something about the biology. But when you have breast cancer and you're triple negative, that means that the proteins that we're normally looking for that would tell us something about the biology of the cell aren't there, right? And so that limits our treatment options.
It's true. And that's just one type of cancer that has 3 major subtypes that all have subtypes in themselves. So another thing that separates a cancer cell from a normal cell is replicative immortality. So they've developed a way that they can always stay alive. So that the normal cells, like people, age, they die, and they have to be.
They have something called the Hayflick limit.
Yeah. Telomeres.
Yeah. So the Hayflick limit is pretty much we undergo a certain amount of cell divisions, and depending on the cell type, the Hayflick limit can range. I think it's like in the 40s or something like that. But yeah, so once the cell reaches its Hayflick limit, it no longer divides. It's stuck in senescence. It can no longer divide. Cancer cells completely obliterate that concept.
And certain types of stem cells in the body don't necessarily follow that rule as well.
Well, stem cells are the only cells aside from cancer cells that can even go above the Hayflick limit.
And that's why there's a lot of theories that, telomerase and like extending your telomeres can give you immortality overall. But
you also get cancer.
You will get cancer.
Yeah.
Yes, you will. You'll be like Deadpool. Good reference, right?
Right. Yes.
But
Deadpool 2 coming out.
yeah, did you see that trailer?
Yeah.
I didn't get to watch it yet, but
No, it's pretty good.
it's
Like
pretty
it.
dope.
Yeah.
Nice. But so basically, nice little sidebar there. We like to have fun over around here. So it gives these cells immortality, but at the same time, it actually enhances not enhances, that's a terrible way to phrase it, but it increases their risk of actually developing DNA damage.
Right.
And actually having, chromosome breaks, double-stranded breaks that lead to more, fragile DNA, more mutations, and the ability of these cells to escape dying, that they'll proliferate and then they'll repopulate, and then that's a hallmark of cancer.
Some really smart guys way back in the day figured out that during DNA replication, bacterial cells, when they divided, they had a certain amount of mutations per million base pairs, or something like that. Per million pieces of DNA that had a certain amount of errors. And for the bacteria, that was cool, because that allowed them to alter their genetics, so to speak, and a lot of times that confers proliferative advantage or survival advantage. Same thing with cancer. Even though eukaryotic cells or human cells have a higher fidelity when it comes to DNA replication.
Fidelity?
Yeah, that's what it's called.
I know, but
Right.
break it down a little bit.
Oh, right. Oh, so there's less. Right, there's less errors per 1 million base pairs when replicating DNA, right? That's one of the good things about eukaryotic cells, humans, animals.
There's more checks to the systems.
Right, so when cancer cells proliferate more often than normal cells, that rate of error doesn't change, but they're proliferating more. So there's bound to be more errors on average.
Yeah.
And that's how more mutations occur on top of genetic instability and everything else that we're going to get into. But that's how cancer cells can continue to mutate over time.
That's a very good point. And actually, I actually just looked at my notes here, and I had a good analogy about telomeres. Telomeres are like the fuse to dynamite. That if you start getting shorter and shorter and shorter and shorter, once you get down to the end, boom.
Somebody actually, back in undergrad, this must have been like, we were talking about telomeres, and they compared telomeres to like the plastic part of shoelaces.
Yeah.
Like the end of the shoelace.
All right, that's so good.
Yeah, because telomeres are actually protective, right? And this goes back to like super basic biology. Chromosomes are linear.
Telomeres are the ends of the chromosome. So
Right.
I think we should have said that at the beginning of it.
Right.
Oops.
Yeah.
But yeah.
You dropped the ball there.
Oops. But yeah, like Lee was saying, the ends of the
Right.
shoestring.
So because we don't have a circular genome, there's something called this end replication problem that the earlier eukaryotic cells in evolution had to figure out how to deal with, right? So because the chromosomes are linear, when you make a copy of it, you lose some genetic information. So all those cells that couldn't fix that died. But the ones that developed this technique to deal with that problem is they started making random, I don't want to call it random because they're a specific sequence over and over again that when you lose that genetic information, doesn't matter, right? So you have core genetic information, let's call that the dynamite part, and then you have the fuse, which is the telomere,
Yeah.
right? And so you make a whole bunch of this, but over time, this fuse gets depleted, right? And when you get to the dynamite part, which is your natural genetic information, you start to lose stuff, and the dynamite explodes. Essentially, the cell undergoes cancer.
Boom.
Hey!
And boom goes the dynamite. All right.
We need an animation for
I
that.
know.
Can we get a video? Can some arts person please, like,
We're looking for
this and that? Yeah.
We're
We're looking for
interns.
interns, please, because we need some animation stuff going on for these explanations.
Yeah, and I'm not the greatest at PowerPoint. I'm getting better.
Yeah, we're getting pretty good. We're starting to make figures now. It's painstaking.
Sucks.
Yeah.
That's why
we're getting good though.
That's why I have a computer that can draw stuff instead
Right.
of having to do all the parallelogram
Yeah.
and this, that, and the other thing. But okay, so the next hallmark of cancer is inducing angiogenesis. And what that means is when you have all these cells rapidly growing and dividing and filling up a sack of cells, like this is literally what a tumor is, you need to get nutrients into the cells, into this tumor, and you have to get the trash out of the cells. So basically, I related it to a new development where you have all these new houses being built and you need a road to get in and a road to get out. So that's basically what a tumor does. It sends out signals such as VEGF and what is the couple other ones?
Ang1.
Ang1, angiopoietin, and then not
It
signaling.
represses stuff that blocks angiogenesis, like TSP1, which is a negative regulator of vessel growth. But pretty much those are all ways to modulate how blood vessels go. Now, I want to make a distinction here because angiogenesis is not growing new vessels, it's bringing existing vessels towards you, right? So the tumor cell is here and it's sending out signals.
It's not vascular genesis.
No, vascular genesis is actually
Maybe.
growing like
No.
from scratch, like raw material creating blood vessels. Angiogenesis is taking a network of blood vessels and causing those blood vessels to come in and
Similar to like
intertwine
how,
with the tumor.
like similar to how a development builds off the existing infrastructure,
Right.
such as a road or a highway, and you have a road that goes down into your development.
Right.
And you start building and building and building. Now imagine if you had one road in that development and you made, 1,000 homes.
Right.
You couldn't get to everything. So that's why the tumor has the ability to grow these. Like basically, think of them like maybe like a plant, like just growing like a set of vines into the tumor.
Yeah. Well, the plant would be the blood vessels,
Yeah.
and then the tumor would be like
Air.
the squirrel.
The squirrel.
Like the squirrel would be like whispering to the tree, and then all of a sudden the tree would extend a branch to the squirrel for it to climb on. That's kind of like what's happening here.
That's pretty
And then the squirrel would then use that tree branch to get to like another tree, and then that would be the metastasis.
Yeah, that's true.
Yo, that's it. Squirrel tree reference
Squirrel
for,
trees.
for angiogenesis and metastasis. I said, wow, this is crazy.
So there's also the flip side to it, though. Remember how we sat through the Dr. Winter, not Jordan Winter, not Ed Winter, where he was talking about a land of not plenty in pancreatic adenocarcinoma.
Oh,
So in certain
is
cancers,
this the NIH guy?
no, he's in Brody's lab.
Okay.
So basically, he's studying
Oh,
a
that
type of
guy.
cancer
That guy.
where, yeah,
Gotcha.
Chris. So this gentleman is studying basically pancreatic ductal adenocarcinoma, and all that means is that's the most awful type of cancer you can get. But you can have an area where you don't have a lot of veins, arteries pumping blood in, pumping blood out, and you have a hypoxic area, and these
Which
cells
means low oxygen.
Yep. And low nutrient levels. So these cells can adapt to that type of environment and proliferate and grow and actually outcompete the cells that need more oxygen and more nutrients, and the flip side to it. So it's not just one thing fits all.
Yeah, of course. It depends on the cancer, right? Because part of the reason why, part of what we talked about last episode with the Wienberg effects, a lot of it has to do with the toxic environment and it being able to modulate its metabolic demands based on the oxygen and nutrients available.
And basically, the way that I had it in my write-up is, one cancer's desert is another cancer's paradise. So think about it like that. So certain plants and animals live in the desert, certain animals and plants live in the tropical rainforest. Just like cancer, you can fit all those niches. And that's why it's damn near impossible to have a cure. So
When I cure cancer in 2082, I'm gonna take those words. No, stop.
It's gonna be alive in
Yeah.
2082.
I don't know why you realize how far away that was. So I was like, wait, how old is this guy?
100.
Yeah, I'll be like,
90.
no, wait, born in
92.
I'll be, yeah.
Yeah,
Anyhow.
you'll be old. Okay. So the next section is activation of invasion and metastasis. Oh, I didn't put of in my header. Yay. That's what happens when you don't proofread your stuff, Lee. So there's a couple of things that can happen with cancer cells. So you have a primary tumor, where let's say it's lung cancer, prostate cancer is a good one. You have your tumor develop in that particular area, and then most people know, I assume that metastasis is growing that tumor in a different part of the body. And that's how most people die in terms of the complications of their cancer.
Yeah, most people actually don't die from the primary tumor, with the exception of some brain cancers, just because you have the least amount of room to spare associated with those cancers. But yeah.
And basically, you go through a process. So you have your primary tumor, and then you have pioneer cells, pioneer cancer cells that go through what's called EMT, or epithelial to mesenchymal transition.
Mesenchymal?
Mesenchymal? Yeah. See, he says mesenchymal, I say mesenchymal. It's potato, potato. He's potato, I'm potato, so I'm right. Because who says potato?
Tomato, potato. Who knows? First, let's just like distinguish what epithelial and mesenchymal cells
So
are.
epithelial cells are like what's in your throat, your stomach lining, your nose, your skin is epithelial cells, and mesenchymal and mesenchymal cells are more stem cell-like, or they have the ability to move and grow and invaginate, what's called invagination.
Should they have more
extravasation.
morphological dynamism?
Yes, that's great. I'm trying to be simple here. It's like morphological dynamism.
Yeah.
Is it bougie up there? Is it like?
I don't know. I need a latte right now after creating that word. I need a 2 sugar.
So basically, what Lee is going through and saying is that the signaling around the tumor microenvironment allows these epithelial cells to become mesenchymal, or they get the ability to change their features so that they can migrate and move and go to different areas. Like we were talking about contact inhibition.
Right.
Mesenchymal cells don't have contact inhibition, or they've lost the ability, or they've
Well, there's some free-floating cells and some microglial, some neuron-associated cells that aren't necessarily contact-dependent. But by and large, most cells in your body are composited. They're part of bigger tissues. Mm-hmm. So freeing them up causes them to die, right? But for example, your entire immune system is floating around your bloodstream, right?
Yeah.
So they don't necessarily need to be contact-dependent. They're like kind of like a unique case. It's always like unique cases.
Yeah, but like your liver cells, your prostate cells.
Right,
these
you know,
types of
hepatocytes,
cells,
you know.
They need to be touching each other, and they also need to be touching the foundation. So what's all called the basal membrane, or the basal lamina, or the extracellular matrix, whatever, all those terms. And if you lose contact with that, the cell should die. But these mesenchymal cells have gained the ability to lose that contact with the surrounding environment.
By downregulating adhesion molecules or
Mm-hmm.
pretty much.
Integrins, cadherins.
Imagine if
these things.
there were a whole bunch of me, we were all wearing this blue sweater, and that blue sweater allowed us to touch each other. Well, if I was a cancer cell and I was like, you know what, fuck you guys, I'm gonna take this blue sweater off.
Be an
And
orange
yeah,
sweater.
that's it.
Be
I'll
an orange sweater,
go
so
put
you
on
know
another, then I get to parlay with the orange sweater guys. But that's kind of how it works, like in simple terms. The cancer cells change their ability to associate with other cells, and that leads to metastasis.
And basically, those cells that, like we said, when he changed the sweater from blue to orange, had the ability to go out through into the bloodstream or the lymphatic system and find a new area to like live. So think of it as like, you know, a colonizer from the Europe coming to America and founding a new
Some
society.
horrible references because we know what happened there. Anyhow,
It's cancer.
yeah,
dude.
exactly.
Like,
That's exactly what happened. Settlers came over and killed a whole bunch of people. It's just like what happens with cancer.
Yeah, I thought it was pretty accurate.
Yeah.
Sorry, it's a dark topic.
It's a dark, yeah, history is a lot darker than people make it seem. It's
So is the
People suck. But So basically, these pioneering cells can go to a different area and start growing what's called a secondary tumor.
Right.
And
It's manifest destiny all over again.
oh God.
Anyhow.
Taking what you want. Taking what you want. No repercussions. So in prostate cancer, a lot of the sites for secondary metastasis are bone, lung, liver, and there's certain cancers that go to different areas.
Some of this is based on proximity. A lot of it is based on the signaling associated with these cancer cells. Actually, where tumors decide to metastasize is highly complex. It's a field of study all on its own.
Yep. And basically what we mean by metastasis is when you start getting into the higher stages. So stage 2 is, stage 1 is that you're just
Primary
contained
tumor.
in the primary tumor site.
It's got
You
a
have it-
certain diameter.
Yep. Stage 2 normally means that you've gotten to the basal membrane, so the layer that surrounds every organ,
Lymph
basically.
nodes.
That's stage 3.
Yeah.
Yeah, stage 3 is lymph nodes. Well, you can have 2A and 2B.
Right. Like 2A. Anyhow, listen, there's stages of cancer, and they're based on from getting from point A to point B. Point B is another tissue or organ, right? So if you got butthole cancer and you make it to your liver, this is stage 4. This is a stage 4 clanger over here.
Stage 4 clanger.
This is bad news. You know?
This one from morbid to like
Yeah.
weird.
Hey, shout out to all my stage 4 clangers. You know, do some references too.
Perfect. So what other things do we plus?
Wait, we got to get to genetic instability.
That's coming up. All right. So in 2010, the same group, Douglas Hanahan and Robert Wienberg, put out another paper, Hallmarks of Cancer, Generation 2, where they put two emerging hallmarks and 2 enabling characteristics. The 4R, deregulating cellular energetics, just like we had said about the Wienberg effect. Avoiding immune destruction, so basically your immune system keeps a lot of cancer cells in check. So that's why there's a lot of immunotherapies like PD-L1,
Keytruda.
Keytruda.
Keytruda actually helps your immune system hone in on the cancer cells.
Bingo. And then there's what? Genetically engineered.
Chimeric antigen T cells. Pretty much, you take T cells from your body, engineer them, and put them back so that they have essentially special spectacles that allow them to see and target cancer cells. But
we make them better detectives to go take out the cancer cells.
and give them rifles and snipers and stuff like that. Yeah.
So that doesn't work for every cancer. That works for mostly.
We arm the rebels, essentially.
We arm the rebels. I love it. We arm the Native Americans. It's kind of like France versus England. God, I love history.
Yeah.
So the next thing is tumor promoting inflammation, and then your favorite, genome instability and mutation.
Right. I just want to talk about the whole immune evasion thing. So when
Do you want to talk about that when we get to that slide?
never mind. I will let Dave talk about programming hallmark capabilities by intracellular circuitry.
Yeah. So basically, how we said before that you have different proteins at different nodes. So you have RAS here, and let's just go through this little signal. Protein talks to RAS talks to the next node, the next node, next node. You have
Professor
divergence.
Davis.
So basically, what happens is circuitry gets hardwired or rewired into thinking that everything's hunky dory and we need to keep growing and growing and growing, and that changes gene expression, gives you different hallmark capabilities such as changing your energetics and the way that you get energy, and the ability for the cell to say, we're in a bad environment, however we're going to live in it. We can adapt to it. It's like being outside in the cold without a jacket, and all of a sudden, I don't care about the cold anymore. You don't get sick. I wish my body could adapt like that. But then I'd feel like I get cancer. But I'm also already really hot when I'm outside. But besides the point. So basically think of the cell as a series of switches and networks where everything's interconnected, and this is such a simplified diagram that all these little green dots can actually be tied back to each other and be tied to other green dots that we haven't even discovered yet.
Yeah, this is kind of like giving me anxiety from Biochem 2 all over again.
But I know that this is something that you wanted to talk about in particular, genomic instability and mutation.
So I'm going to give you my version of this because there's just a lot going on in that slide. Pretty much, you got all the cells in your body have the same genetic material. When you get mutations, meaning you change the spelling in the genetic information, and when you change the spelling, you get different proteins than you needed to before. And by genetic instability, we also mean by chromosome translocations. You stick chromosomes together, you delete whole parts of chromosomes off, you flop them, you swip, you name it, it can happen in the world of genomics. All these things can result in cancer. And one of the hallmarks is genetic instability because the cell is rapidly dividing. And it also upregulates proteins that create more genetic dynamics that allows these mutations to take place. Like replication stress, or it's just a lot.
Yeah, it really is. And by having genome instability, you get more mutations, and then you can actually have what's called microevolution, quick evolution, where if you get a mutation in one cell out of like 100 cells. Let's say the tumor has 100 cells, which is not true. It's gonna have thousands of
billions of cells. Yeah.
And
tumor heterogeneity.
Yeah, exactly.
That's such a hard word.
Tumor heterogeneity.
Heterogeneity, yeah.
Heterogeneity.
Heterogeneity, yeah. Pretty much, if you got a soccer ball.
You
you got a soccer ball. It's got is those hexagons on a soccer ball?
I think so.
Somebody Google that, please. I think there's hexagons on the soccer ball, and some of them are black, some of them are white. Pretty much the tumor's like that too, right? But imagine if the black ones and the white ones had different genetics that offered some type of pro-survivability for that tumor, right? So you get a drug that kills the white cells, and then the black cells just recreate the soccer ball. Now it's not a soccer ball anymore.
It's called a truncated icosahedron.
Is that what that shape is in a soccer ball?
So there's 12 pentagons and 20 hexagons.
I'm almost certain whoever invented the soccer ball did not know that. I can guarantee that.
I think it was just a leather ball at that point.
Yeah, I'm pretty sure soccer was like prerequisite in like I'm pretty sure like it was made out of like pigs or something. Like there's no way they figured this out. What was that called?
Icosahedron.
Yeah. Anyhow,
I can't even say it.
A lot of retroviruses have icosahedron capsid proteins actually.
See, there you go.
Yeah, I knew what it was. It was just weird that that's what that shape was in a soccer ball.
Yeah, nice. Soccer is like a virus. Okay. So did we get the point across about how the black one, the little...
Yeah, so yeah, you get tumor heterogeneity, yeah, so you have different populations of tumor. So yeah, that changes the way we can treat things. That's part of the problem to begin with.
They
but
can
yeah.
accumulate new mutations, each individual one, and they can pass it on to their descendants or the cells that they divide into.
Their progeny.
And then they get more mutations, and that'll give them a leg up to outcompete the other cells. And it's kind of like coming back to the point where if you blast it with some type of treatment, the cells are going to adapt and they'll outgrow, and they will basically beat the treatment. And it really comes down to
so most people get cancer. Cancer is really a disease of old age. Most people get cancer. The median age for cancer, I said, is like early 70s. Even though there's
women have a 1 out of 8 chance in their lifetime, and men have a 1 out of 7 chance
of getting any cancer.
Any cancer.
I thought it was higher than that.
1 out of 7.
It was like 1 in 4.
Maybe.
Because prostate and breast cancer are like
1 out of 7 and 1 out of 8. Yeah. Then it's 1 out of 2, and then 1 out of 3.
Right.
Yeah. I think 1 out of 2 for men and 1 out of 3 for women.
Right. All right. Lifetime risk.
Lifetime
Yes,
risk.
from birth to death, lifetime risk. I think that's about right because the older you get,
It doesn't necessarily mean you'll die from it. It just means
you get. Yeah. I mean, a lot of people get. Actually, cancer death is on a decline. We keep finding ways to punch cancer in the face.
And then it comes back and kills me.
The problem is we need to find ways to stop people from getting cancer, which
Preventative care.
yeah, kind of.
Don't drink, don't smoke, work out. Don't eat fatty foods.
I love Chinese food, but here's an interesting fact: some of the oldest, some of the oldest living people on the planet have moderate alcohol intake in the form of wine and things of that sort. So everything is really kind of dose dependent thing, and
It's all relative.
So yeah, it's
What works for one
depends.
person doesn't work for another.
Yeah. There's obviously bad things like snorting cheeseburgers for breakfast for the next 15 years of your life.
Is that something I can do? Can I snort a cheeseburger? Is that possible?
I mean,
do we have the technology, Lee?
Yeah, we definitely.
I need more energy. I need more food intake. I want cheeseburgers.
I'm almost certain I saw someone do it once. But anyhow, that's besides the point. The point that we're trying to make here is that cancer is really a disease of all age, and most people will probably get cancer if you live long enough. There's so many factors that contribute to you getting cancer. Your immune system is one. Even if you, first of all, you get cancer cells all the time. Your immune cells, natural killer cells and T cells, destroy these cells on a regular basis because, as I was talking about earlier, wait, did we get to promoting inflammation?
Yeah.
All right, we're going to get to
promoting inflammation right here.
So when a cell turns cancerous, normally there's some stress indication before that, right? They start expressing stress-related proteins. And immune cells can pick up on that. They bind to them and they usually kill them when they're expressing stress markers. Well, I'm on the wrong slide because I'm talking about immune evasion.
Yeah, we were talking about that.
Did we talk about immune evasion?
Oh, no, it's coming
It's coming up.
All right.
I'll let you talk about inflammation.
So tumor promoting inflammation is that you go through apoptosis or you go through necrosis, that pathway, and you release a whole bunch of cytokines, which are signaling molecules to the environment, and that makes the tumor actually more aggressive. It actually signals to the other cells, hey, something's wrong. We need to reprogram ourselves, rewire ourselves, and go, nope, we're not going to die, and we're actually going to grow quicker and quicker and quicker. So that's basically what that means. Wienberg effect, reprogramming energy metabolism. Basically, you can have an area of low nutrients, not a lot of sugar. That's basically glucose is the thing that we all run on, literally, like Duncan. Yeah, it's terrible. I know, I'm not fun. So it just rewires the cell to use different means. So I know that we talked about this 2 weeks ago when we had the metabolics and metabolomics, and
right.
Where you go from oxidative phosphorylation using the mitochondria, the powerhouse of the cells, and going through what's called glycolysis, and that's basically just splitting glucose into two parts and using pyruvate to go to lactate. As everybody knows about lactic acid and feeling the burn when you work out because you don't have enough oxygen in your muscles.
Aerobic glycolysis, yeah, pretty much.
And the tumor cell basically rewires itself and gets energy in this means. So it doesn't become that, it's not as dependent upon the normal mechanisms to get energy.
Yeah, also they use glutamate as a precursor for acetyl-CoA and using fatty acids and other type of molecules to plug into various forms of energy-producing schemes, so to speak, or pathways. The cool thing about, I won't say the cool thing about cancer because it's a shitty thing, but it is cool to study it. The cancer cells have all of the genetic information at their disposal, right? So when neurons actually use more glucose than any other cell in the body, but that's because of the genetics that they're using at that moment, right? So the cancer cells, because they have this genetic instability, they're pulling on pathways that other cells normally use only in certain circumstances, right? So they're like, oh, we have this plethora of molecule here. Well, let's use this area of the genome that codes for genes to, use this as energy. Now, the cancer cell really isn't making that choice. It's the population of cancer cells, right? And because that is their environment, everything else dies and takes a shit, and the ones that just happen to be using that genetic information will repopulate the tumor cell. Yeah,
it's basically random chance that if you have the information available to you, you will use it.
Right.
And that's how these cancer cells work.
And it's a numbers game, right? And there are definitely stimuli associated with the cancer's ability to use that, and then the environment plays a role. So yeah, it is a numbers game. The more cells that are doing different things, the higher chance you get of some of those cells doing the right thing that allows them to survive.
All right, here we go.
All right, mating immune destruction. All right, so pretty much, here's this cool thing. When I was studying natural killer cells would bind to tumor cells via these protein interactions like Dynam-1 and stuff like that. And then the cancer cells would secrete enzymes that would chop off the receptor.
Nice.
Yeah, and so then the natural killer cell would disassociate from the cancer cell. I was like, that is swag.
That is swag.
That is so crazy.
It's like, you're going to try to kill me?
Yeah, exactly. Like, oh, your mind was up there. Boop, boop, boop. Yeah, I was like,
Sound effects are great.
I thought that was so crazy for the cancer cell to use that as a mechanism for.
They do that with the other white blood cells.
Right.
T cells, cytotoxic T cells, dendritic cells, macrophages. And and a lot of things that they can do is they can pump out cytokines like IL-6 or IL-10, or they can actually bind halfway to like T cells and cause T cell anergy. What T cell anergy is that it just deactivates the T cell. So instead of becoming a killer cell, it's just floating around and it's.
Yeah. They also recruit suppressive immune cells like T regulatory cells called Tregs. There's something called FOXP3 positive. Tregs for short. So inflammation has this pro and con type thing. Obviously, you need inflammation. When you fall down, you sprain your ankle blows up because acute inflammation is good.
Yep.
It gets blood cells there. It causes vascular permeability. So you have type 2 macrophages going there and repairing whatever damage. There's a lot of things going on with that approach. Chronic inflammation or sustained inflammation is detrimental.
Yes.
And cancer cells kind of play this balance between modulating the inflammatory state in a local environment for their benefit, right? Because causing inflammation necessarily brings more immune cells towards you.
Yep.
That's a bad thing, right? Immune cells are there to kill you. But the cancer cells have found a way to create this homing mechanism, but then once the immune cells get there, they're evading them. Mm-hmm. So they get the pros and the cons.
They can shut them down, they can pump out the signals that actually go, nope, you're not going to actually kill me. So,
yeah.
And that brings us to cancer stem cells.
Shout out to tumor immunology, which is a whole field of studying this interaction.
I can't wait.
Like people get PhDs in tumor immunology. That's like it's bananas.
Actually, my PhD, what I'm thinking, hopefully my project's going to be is immunomodulatory. So maybe, depends. We'll see how the vasoconstriction goes. But so cancer stem cells. These are basically cancer cells that can actually. So remember how we're always saying that.
Some people don't believe in cancer stem cells. Who said that?
Some people don't.
Some people don't believe in cancer stem cells.
It's a point of contention in the field, and we don't actually
debate
it. However,
I'm not a fan.
I'm a fan. I think that in certain cancers there are, like glioblastoma, I really believe that there are cancer stem cells.
There are definitely some data that supports it, but I think it's more niche.
I guess. It depends.
It depends, right?
Yeah. But with anything, it's not a set in stone thing. But what I really believe is that these undifferentiated cells, in particular like neuroendocrine prostate cancer, you can have these stem cells rapidly dividing and proliferating, but they don't have the normal wiring that all the other cancer cells have. They have the ability to just repopulate tumors very quickly and rapidly. They're resistant to treatments, or they become cancer stem cells based upon treatments. And they can repopulate the tumor microenvironment after a course of treatment and refill up the tumor. And then they can actually go and be colonizers in secondary metastatic sites. Yeah. So whether you believe in the theory or not, it's definitely an area for future investigation.
Oh yeah, of course.
And another point to prove in that there's not going to be a cure for cancer.
Right. Let me clarify when I say that. Some people don't believe in cancer stem cells. The data associated with cancer stem cells is not super clear. And so because of that, there's room for different perspectives. By no means is the topic of cancer stem cells like pseudoscience or bullshit. No, hardcore scientists are doing experiments to see if these things exist and whether we can target them as a way to deal with cancer. Mm-hmm. I think that they're just other types. They're just cancer cells with some strange phenotype. I don't even think they're like a thing on their own. We'll see.
We will. To be continued.
We'll see.
We'll see. But basically, the last slide we got here is just progression and malignancy. So you start off with your cancer cells, you get some changes, they go to that EMT that we were talking about, epithelial and mesenchymal change, they move on to a different site, and they grow again. So that's basically when you have, end of life type of circumstances in tumor with cancer patients. It's the progression to the ability to extravasate.
Extrapolate.
Extrapolate? Don't you do that with data?
No, that's data. So basically, you gain the ability to leave the primary tumor site and go somewhere else. Whether that's in the lymph nodes, the bloodstream, you ended up in the lungs, liver, bone, anywhere that you can get sponged up and then you regrow. And at that point in time, that secondary site can also have those tumor cells grow,
Right.
and then they can leave that secondary site and do a tertiary site and a quaternary site, basically third and fourth.
And the quinterary,
sites. I don't know.
I don't think that's a thing, but 5 times is worse than 4. But I wanted to get into, we didn't really talk about how cancer cells actually kill people.
It's terrible.
So, right? Not jump shots, okay. Cancer cells actually kill people, not because the cancer cells are like producing a substance that's toxic for you, or like bacteria or something like that, or the cancer cell is really growing in a place where it shouldn't be, right? And so if your brain is very compact and organized in such a way for it to operate in X way, when you get a mass that is starting to push on things it shouldn't be pushing on, the cells around it die.
Or if you're like in the lungs, you won't be able to breathe.
Right.
Or if it gets to your heart stops beating. Or if it grows rapidly, puts pressure on the heart, stop the heart.
Muscle cancers are also super rare, by the way.
Sarcomas?
Yeah. Yeah. Well, sarcomas are soft tissue cancers.
I think so. But I think sarcomas are...
They incorporate the muscle as well.
Yeah, and bone.
And bone.
Yeah. Muscle, bone, sarcomas. Yeah.
Right.
Sarcomas, yeah, but they're rare. And that's because those cells...
Carcinomas are epithelial.
Carcinomas and adenocarcinomas,
Right.
ductal.
Right.
And basically, those cells don't rapidly divide as much as, say, your epithelial cells. So a lot of the reason why you get throat cancer and lung cancer is because those cells rapidly divide all the time.
They're being
so
replaced,
therefore,
yeah.
you have a higher chance of getting genetic mutations just by sheer numbers.
Well, people mostly get lung cancer through environmental stimulus like smoking and things. Even though some people get lung cancer, even though they've never smoked before in their life, those people...
It's actually because of the cytochrome P54 pathway. So when you actually smoke and you have a very active
Oh, did you say cytochrome P54?
P450?
P450, there we go.
Sorry, P450. And you have a very highly active P450 pathway. You actually increase the amount of metabolites that cigarette smoke gets broken down into, and those metabolites are actually things that cause cancer.
Just to be on the same page here with our viewers, cytochrome P450 is an enzymatic pathway associated with metabolism.
And it involves iron as the reactive metal. So I just want to throw this one last little conclusion out before we go into our ending, is that cancer is like understanding cancer is like taking somebody from Russia, sticking them in Florida with one piece of paper and a ballpoint pen, and saying, map the entire United States inner system by foot. That's basically think about it, because you have to map every single pathway that is in a cell or could be in a cell, and then you have no...
That is like the hardest scenario ever in life.
Well, think of it. Yeah, that's why there's thousands.
Yeah, yes, you're right. Yeah, I just
toiling away at this.
that was just like, I don't think I could have thought of anything more difficult than doing that.
Yeah. You can't speak English, you can't even talk to anybody while you're walking the highway.
By foot.
By foot.
Yeah.
So that's basically why there is no cure for cancer. And just the complexity, the sheer complexity of the disease just dictates that it's going to be a long while till we figure it all out. And that's why Lee and I are in the field that we're in.
Doing the stuff we're doing. The best hope that we could probably hope for is that we develop enough therapies. One, we develop better diagnostics that show us that people are going to get cancer earlier, right? Because dealing with stage 1 cancer is easier than dealing with stage 3.
Higher resolution imaging.
Right? Detection, biomarkers, all those things will help us identify cancer sooner and thus have better patient outcomes. 2, dose. Numero dose. That's 12.
Dose.
Yeah, just dose. Dose, right?
Dose is 12.
I don't know. Dolce. Yeah.
Dolce?
Dolce de leche is really good.
This is.
Yeah, number 2, making better therapeutics that are selective for cancer cells that don't kill normal cells. The closer we get to that resolution, the less side effects and the less treatment-associated death we'll have. And number 3, understanding the whole environment human interface that allows us, such as smoking and what role diet plays and all of these other environmental stimuli, understanding that dynamic better will allow us to maybe eliminate more preventative type things for cancer.
All right, cool. So thanks for joining us tonight, everybody. That is What is Cancer? In perspective of the...
All of these notes are going to be in the comments. We're going to give you the links, yeah.
I already posted the comments to the Hallmarks of Cancer. So if you want to do a little bit more reading, have better analogies, it goes a little slower.
Yeah.
And there's actually 4 parts to it. So it's quite a read, but at the same
It's
time...
on our website.
Yeah, bioslifemedia.com. The links are in the comment section, so check it out. And let us know what you think. Share it with your friends that think that there's a cure for cancer out there.
Right.
I'd really like to get their perspective. No. But we'll be putting out a new schedule coming up in the next couple days, and I believe, do you have any ideas for shows? I think I want
Some kid recently had some form of gene therapy, and now he doesn't have sickle cell anemia anymore.
that'd be a cool show to do.
That's crazy because there's this big debate about gene therapy, which I'm a fan of, even though people have died. But I think it's getting better, so we should talk about it. Maybe get some backdrop on gene therapy and talk about the stuff that's happened in the past.
Yeah, all right, cool. We'll do that. And then put together a list, and then we'll send it out to you guys. All right, everybody. We'll see you guys next week. Bye-bye.